Great, thank you, Jerry, and thank you all for joining this afternoon. I'm really excited to present this research and kind of introducing this topic around precision feeding alternative forages, and in this case, to cows in their early lactation. And so today we're going to talk about just briefly the aspect of forage planning and how we can apply feeding nutrition practices around cover crops and alternative forages, and then going into a specific study where we utilize these forages in early lactation diets and some conclusions and future potential around this area. And so, as I mentioned, I think that we talk about precision nutrition in a way typically when we talk about automatic milking systems or AMS herds, and really that pertains to grain feeding concentrate mixes to hone in on feeding small groups of animals to meet their optimal nutrition potential. However, I think this can continue to expand into forage planning. A great example of this is feeding brown midrib corn silage versus conventional corn silage to different groups of animals to support them at their stages of lactation. Also, again, these forages provide, looking at the cover crops, they provide another alternative for precision feeding. And whether or not we're double cropping, but knowing that, with some of these double cropped forages, we may be having less tonnage. And so, again, having specific groups of animals that we can feed these to really provide us the opportunity to apply some of these practices. And so with that, I'm really going to turn it over to my master's students that I am advising alongside Dr. Heather White at the University of Wisconsin-Madison, who conducted this project as part of her master's, and let Barbara Dittrich here take it over.
Thank you, Lizzy, for the introduction. Hello, everyone. I'll be talking about the use of alternative forages and early lactation diets. So the study we conducted happened last summer. It included 80 multi-parous cows in early lactation. We monitor these cows from calving up until peak milk lactation, with all the cows starting on a common covariant diet, which is our control diet, which is corn silage and alfalfa silage. And then when they hit that outside of that transition period, hitting that 23 days in milk, we introduced the cover crop silage at a 10% inclusion rate of dry matter in replacement of the alfalfa silages. I do want to make a note, these are the same cover crop silages that were used in Ken Kelcher's study. And this is kind of just an outline of my project and some different feed samples that we collected throughout the project. The red representing the covariant period, where they were fed that control diet, and then the green and the blue represent their actual trial period, with the green representing where the cows are in days in milk, and the blue representing the week in lactation the cows are at. I'm really going to focus in on what we've so far analyzed, which is their dry matter intake and our performance metrics as a milk yield, fat, and lactose, and energy corrected milk. I do want to make a note, we are going to look at some of these other aspects of whether change in body condition, some milk fatty acids, and see the metabolites of the different cover crops, as well as the microbiome. So just kind of going on an overview of the nutrient composition and comparison, we wanted to include that alfalfa silage on here and show the main differences when formulating these diets, what we have to consider. So you can see that crude protein here, that it's at 23 or almost 24% versus the cover crops are around that 12%. And then we also see a big difference in forage NDF, with alfalfa about 47% and the cover crop silage is approximately 74%. And then I also do have included on this slide the harvest dates of when those cover crops were harvested. Then going into the actual ingredient composition of how we formulated the diets and up to make up. For those differences, we really aimed for a 30% NDF, a 26% starch, and an 18% crude protein. Here you can see the differences with that canola meal, higher in the rye and triticale, or in the cover crops. And then in makeup for NDF, we included the inclusion of cottonseed. We did also analyze metabolizable protein, which is why our mono diets have a higher inclusion of canola meal compared to the three-way cover crops. This is our overall predicted nutrient composition, as this will be something we'll have future analyzed, hopefully coming around May or June. Seeing that we balance for metabolizable protein about that 10.7%, that nutrient determinant fiber came out to be about 29%. And then overall, we have the conducted dry matters, and these are the results that we had of our study. Overall, pretty similar, with a little more variation with the control. So our overall results that we saw with our milk performance, we did not have any differences in dry matter intake, which, as Ken mentioned earlier, was a reflection, or it tends to be a reflection in milk production. However, we did see differences in overall milk production across the treatments. One thing I do want to consider that I have on this table is we did an overall treatment difference, but we also did a treatment by week interaction. With early lactation, we wanted to see the lactation persistency and whether or not the cows were performing equivalently across at certain time points, or if we saw positive or negative effects at certain time points. And I have some graphs demographing this in the next slides showing that interaction. The other things I want to make a keynote on is just the fact we saw an interaction there across treatments, an overall difference in protein with the cover crops, specifically with the rise tend to be a little lower than the triticale, and then MUNs, we saw that interaction with the treatment by a week as well, and an overall treatment means. Going further into components and feed efficiency, other things that were kind of interesting that we saw from our results was we saw significant difference with lactose yield, with the rise having higher lactose compared to the triticale. And then energy corrected, or for feed efficiency, we saw that treatment by week interaction where certain treatments performed better or than others, and just that kind of crossover of where we're seeing the results. So here's the graph of our average weekly dry matter intake. I want to make a point out that the black represents the control, the red represents the rye, the blue represents the rye three-way, and the green represents the trit, and the yellow represents the trit three-way. And the bottom represents the week of lactation over the dry matter intake of pounds. And I want to make a keynote. Our study did start at the week 4 of lactation, approximately 23 days in milk. And as you can see, looking at this graph, you have that consistency of dry matter, and all the means stay relatively the same, which we saw no significant difference across the treatment groups. Then when analyzing weekly milk yield, we really can see and visualize the differences across the treatments, with the rye having much higher production compared to the triticale. However, it wasn't significantly different from the control. The only significance we had was from the trit three-way and the rye three-way. I also do want to make a note, all the cows were randomly assigned to the treatments based on when they were calving date. However, each of the groups were balanced for ME305, total lifetime fat production, and lactation age. Then we go into milk urea nitrogen, where we see that interaction again, kind of a crossover with the control, a little more variable compared to the other treatment diets. However, we see that rye appears to be higher in MUNs compared to the triticale. Then we go into feed efficiency, where you really want to account for just the overall what you're feeding for the cow and what results you're getting out of it. One key thing that I really want to point out with this is that the trit for overall means had the highest feed efficiency, even though it wasn't one of the higher producing milk yielding treatments. And as you can see, almost all of our treatments besides the trit three-way appear to have higher overall feed efficiency compared to the control over the weeks of lactation. And just that same similar graph, but with the inclusion of true protein in that production metric. And we see that we have similar results happening in this graph as well. So conclusions and future potential cover crops and dairy lactation diets. Cover crops can be successfully fed at 10% of the dry diet matter in early lactation. Our results kind of showed some differences in milk yield, protein, and fat responses. And then with future feed analysis and microbiome analysis and the blood metabolites and digestibility, we can give a better indication of why we saw these results. So some other knowledge gaps and conclusions. Inclusion of alternative forages and cropping rotations offers some unique opportunity for precision feeding. More data. We do need some more data though, including alternative forages, like how much amount, whether that Ken talked about that 15, maybe 20% in late lactation. Is 10%, the key safe spot for early lactation, or could you increase that? Different varieties, and then replacing maybe corn silage versus alfalfa silage. And then also just some more information on the nutrient analysis of whether it's fresh or ensiled, and what ages of life stages should you be feeding it at. I want to thank Dr. Lizzy French for allowing me to present and be a part of this project, along with Heather White and the Focus on Forage group, to be able to present my research.